that dua which we do, dua mushtaraka Mah Ramadan, it is meant for Allahumma ighfir kull ahl al-qubur, all the people who have died. It's an international dua, so we should pray for everybody. And when we pray for everybody, inshallah, Imam Zaman will come. Inshallah. Fajil dar Faraj. Bismillah ar-Rahman ar-Rahim. Alhamdulillah Rabbil Alameen wa sallallahu ala Muhammadin wa ala tahirin. See, today we have a quiz. Don't get scared of the quiz. The quiz is very easy. I will give you plenty of time for the quiz. You can send it to my email like before. And all of you please write down the quiz, because it will help me to give you marks for your finals. I think about 10 marks are for your class. So about 3 to 4 marks for your attendance. And the rest I've kept for your quizzes. So please, all of you, write your quiz. Do not be afraid. Even if it's wrong, I will give you marks at least you have tried. And it's quite easy, so it's not going to be so difficult. So for the time being, please concentrate on the lesson. It's a very interesting lesson. We talked first, if you remember, in our first class, we discussed proteins, amino acids. Then we talked about transcription. Then we went into chromosomes. Then we talked about heterochromatin and euchromatin in chromosomes. Now today we will go on to the nucleosome. But first, again, we will discuss chromosomes and chromatin and nucleosome. And the best part is that many quiz questions are there in what we're going to do today. So do not worry. Chromosomes are DNA associated with proteins. You remember we said that there are more proteins than DNA. Everybody knows the answer now. The chromosome is a compact form of the DNA that readily fits inside the cell. Packaging the DNA into chromosomes serves to protect the DNA from damage. Only DNA packaged into a chromosome can be transmitted efficiently to daughter cells. So what is the function? Protect DNA from damage, transmit it efficiently to daughter cells. Now, variation in chromosomes in different organisms. The traditional view is that prokaryotic cells have a single circular chromosome and eukaryotic cells have multiple linear chromosomes. Okay, this is your prokaryotic cell. It doesn't have a nucleus. It's got something called a nucleoid. You have your plasmids, capsule. Then you have a eukaryotic cell, a diploid cell, Golgi apparatus, nucleus, chromosomes. This is diploid, this is haploid. Comparison of gene density in different organisms' genomes. Gene density for different organisms. E. coli has got 57 genes. Suppose this is 57 genes of E. coli. Okay. It has got RNA polymerase genes, repeat sequences, introns, genes, intragenic sequences. This is 31 genes. Drosophila, 9 genes. In 57 genes, RNA polymerase genes are little. Hardly intragenetic. Introns also very rare, little. Drosophila, no. See? Lots of introns, lots of intergenic, repeat sequences also. In humans, 2 genes in the humans. A lot of repeat sequences, a lot of introns, etc. Just to show, this is a 65 kb region. So the human genome is 3.2 megabase. Intergenic DNA is 2. Okay, more is intergenic. Other intergenic regions, 600 genome-wide repeats, which are repeated. Okay, we have microsatellite, which is 90 megabytes, and unique 510 megabytes. The human genome, genes and gene-related sequences are 1.2 megabytes. Related sequences, genes, introns genes, pseudogenes. These are genes and gene sequences. So you've got more intergenic, repeats, microsatellites, unique. So this forms genes, these forms genes which get translated. These forms genes which rarely get translated. Pseudogenes. What are pseudogenes? No. What are pseudogenes? What are pseudogenes? Come on, you can Google and tell me, don't mind. What are pseudogenes? Non-functional genes, very good. So he said a functional gene can become non-functional. Okay, fine, very good. Good. It can become non-functional by transcription or splicing. There's a thing called reverse transcription. When it becomes reverse transcription, this gene this way becomes the other way around and it becomes a pseudogene. Okay? So the functional gene can become a pseudogene with the help of reverse transcription or reverse transcriptase. So these are all. So the majority of human intergenic sequences are composed of repetitive DNA. Intergenic, other intergenic regions, dinucleotide repeats, genome-wide repeats, mostly transposable elements. Now, one question comes here. This part is in heterochromatin or euchromatin? Repeat. You remember we did the last time, and this is also part of your quiz questions. Repeat sequences are in hetero or eu. This part here, this will come in heterochromatin or it comes in euchromatin. This part comes in hetero or eu. This is gene and gene sequences which can get. Okay, hetero, not you. Aha, not you, please. Repeat sequences are in hetero because they do not get translated, if you remember. Okay. So, hetero, good. Now, here, gene and gene-related sequences are introns, UTR. What is UTR? What is UTR? What is UTR? Come on. You can Google and tell me, I don't mind. Because I want everybody to be awake. Okay, good. Untranslated region. Good. So this has got introns, untranslated regions, gene fragments, pseudogenes. They all come here. Okay. And genes are 48 megabytes. Of your 3.2, 48 megabytes are the Allahu Akbar. Now, contributions of introns and repeat sequences to different genomes. Now, chromosome duplication and segregation. Eukaryotic chromosomes require centromeres. You remember we did telomeres, and origin of replication to be maintained during cell division. You have your nuclear membrane, you have your telomeres, origin of replication starts here, and your centromeres. Now the DNA is replicating, replicates into 2, mitotic spindle gets pulled apart. This is mitotic division. More or less than 1 centromere leads to chromosomal loss or breakage. Okay, if you have more than 1 chromosome, 1 or more centromere, your chromosome gets lost or it breaks. Here there's no centromere, random segregation. But here, 2 centromeres, again it doesn't break, it doesn't distinct very well. Chromosome breakage due to more than 1 centromere. Centromere size and composition varies dramatically. You remember we said that in the centromere all tandem repeats, a lot of repeats are there. In the S cerevisiae, the fungus, 125 base pairs, S pombe 40 to 100 kilobase pairs. This is base pair, this is kilobase. In the Drosophila, 400 kilobase, and in the humans, 240 to several megabases. The centromere size, okay? Now telomeres. Telomeres are bound by a number of proteins. These proteins distinguish the natural ends of the chromosome from sites of chromosomal breakage and other DNA breaks in the cell. Now, the DNA ends of the this form is wrong. It is from sites of chromosome breakage and other DNA breaks in the cell. DNA ends are the frequent recombination and breaks take place at the end of the DNA. But as we know, the telomeres keep the structure of the chromosome together. They don't allow the DNA to break anywhere at the end. And they recombine together. They do not take part in recombination. This is one of the questions of your quiz, I think. They do not take part in recombination. So because they segregate together. The proteins at the telomeres form a structure that is resistant to both events, to frequent recombination and DNA degradation. Telomeres act as a specialized origin of replication that allow the cells to replicate the end of the chromosomes. Okay? So, we said the specialized region of replication, if you remember. Where was it here? Here, I think, here. Origin of replication, it could be the telomere. It could be anywhere. So they act as specialized origin of replication that allows the cell to replicate the ends of the chromosome. Okay. Chromosome segregation, mitosis. Prepare for cell division. DNA replication. Prepare for chromosomal segregation. Chromosome mitosis. Key events in S phase, initiation of DNA replication. The DNA is being replicated. Each chromosome of the duplicate pair is called a chromatid. The 2 chromatids of a given pair are called sister chromatids. This is cohesion, okay, with the help of the protein. We'll study this in the next slide. Sister chromatids, and they stick together with the help of these proteins. Cohesion, kinetochore, microtubules, pulling the cohesion breaks. They destroy the cohesion. These are cohesion proteins. Changes in chromatic structure, DNA. You know, this is decondensation in the interphase where the DNA is replicated, DNA replication, and then man has to undergo the M phase. Chromosomes are maximally condensed in the M phase. Cystochromatic cohesion and chromosomal condensation are mediated by SMC, structural maintenance of chromosome proteins. So the cohesion of the chromosome and the condensation are mediated by SMC, structural maintenance chromosomes. Okay, so all this cohesion and all that, and the condensation is by SMC, structural maintenance chromosome proteins. Models for cohesion and condensing, these are all SMCs. This is cohesion and condensing. Condensing comes here to condense them, to get them together. This, the cystochromatids are attached here. The structure of cohesin is a large ring composed of 2 SMC proteins and a third non-SMC protein. Then it breaks up. Mitosis maintains the parental chromosome number. We know this, we have studied all this in the last prophase and the metaphase. So the same number will come, diploid, anaphase, telophase. Meiosis reduces the parental chromosome number. Cohesion is lost. Formation of chiasma, homologous recombination. Formation of chromatin structures. Now we come to the nucleosome. What are the nucleosome? Nucleosome are the building blocks of chromosomes. You remember we said like the tasbih, the DNA around the nucleosome, like strings of a tasbih. Histones are small positively charged proteins. The H2A is red, H2B is yellow, H3 is purple, and H4 is green. Now one question I have here. What are the forces interacting between all this? What are the forces in cohesion, in condensing, in histones? What forces keep these things together and separate them? Can you answer me? We have studied this in the amino acid in the first slide lecture. Let me see. Can anyone tell me? Electric hydrogen bonding. Okay, good, good. Hydrogen bonding, then hydrogen, ionic, van der Waals. Very good. Okay, not electric only. Okay, electric is a different thing. I forgot to take the last attendance. Please remind me to take your attendance, okay? In the last class, I forgot to take their attendance. Anyway, I've given them the quiz, so I'll have to know how many have come. So we said H2A, H2B, H3, H4. Now H2A, this is the N-terminal tail, this is the histone fold, the C-terminal, these are all proteins, right? So you have C, H2B, you have H3 and H4. Now H2A and H2B is a dimer, and H3 and H4 is a tetramer. Tetramer means 4. H2, H3 and H4. Together they form an octamer. It's called an octamer. Tetra means 4. So H2A and H2B fold like this, and H3 and H4 fold like this. Now why we are talking? Because when you want, every time, now we were talking about metaphase, and we were talking about synthesis of chromatin, of protein, we were talking about synthesis of chromosome during metaphase and during anaphase and things like that. All the time that DNA is getting replicated, at the same time nucleosomes have to be made, so that the new chromatid goes around the nucleosome, right? Very important. So there has to be an assembly. Each time it gets formed, the histones have to get formed, they get assembled together, and the DNA goes around it. You know, when you look at it and you realize that Allah is such a great, Allah is so great. 1 minute, I'll just get. Okay, sorry. So every time it gets assembled anew, each time. A new sister chromatid gets formed, at the same time the DNA is getting formed, a nucleosome also has to get formed. For the nucleosome to get formed, the histones have to get assembled together, okay? So you get they get assembled like this. Each time, first H2A, H2B gets together, H3, H4, and H2A, H2B. Once they get together, then the H3 and H4 come, and then H2 and H2B come. And that's how they get assembled. And the tails are outside. The N-terminals are accessible to protease trypsin. N-terminals are accessible to protease trypsin. Specifically cleaves protein positively charged amino acid. Trypsin cleaves protein positively charged amino acids. And the nucleosome has an approximate two-fold axis of symmetry. What do you mean by that? One is like this. Okay. Entry and exit of the DNA. Second one is the other way around. See? One is flipped over. You have the entry and the exit. Flipped over. Interaction of histones with nucleosomal DNA. First we have, as we said, the H3 and the H4 region come. Central 60 base pairs at the two ends. Then the H2B and the H2A and H2B dimer come, each associate with about 30 base pairs of DNA on either side of the central 60 base pairs. Okay? 60 base pairs on the two ends, and here with about 30 base pairs of DNA, each of them on either side. Here comes about, I think, 120 base pairs of DNA, if I'm not mistaken. Now, histones contact the minor grooves of the DNA by forming a large number of hydrogen bonds. You remember we talked about the hydrogen bonds, that van der Waals forces, ionic forms. The large number of the hydrogen bonds provide the driving force to bend the DNA. If the forces were not there, it would not get bent. Oh, it's something so beautiful. I mean, when you keep thinking of every cell on the planet, this is happening all the time to it. Allahu Akbar. Now, the H binds to link a DNA H1. Now what happens is, when they are bound, they link a DNA H1. H1 will bind the two together. So you had H2A, H2B, H3, and H4 inside. And when it gets bound, an H1 will come and bind it here. It's like a clip. The nucleosome and the central H1 binds to link a DNA at one end of the nucleosome and the central DNA helix. Okay? The addition of H1 leads to a more compact nucleosomal DNA. Allahu Akbar. See, this is without H1, and this is a picture with H1. H1 induces tighter wrapping around the nucleosome. Now, this is good in certain conditions, especially when the cell is dividing. But when the cell has to be replicating, the DNA has to replicate and translate, you don't need such tight binding. So something has to happen to prevent that. Now, for the binding with H1 binds like this, and the histones can become solenoid, means very tight, 6 nucleosomes per turn. 1, 2, 3, 4, 5, 6. 2, 3, 4, 5, 6. This is called solenoid. If you cut this in the middle, you will see. This is called solenoid. And this is the linker DNA. This is your EM and X-ray studies. There's another one, zigzag pattern. When H1 is added, it requires linker DNA to pass through the central axis. Okay, is that clear? So what is linker DNA? Linker DNA is a double-stranded DNA, 38 to 53 base pairs long between the two nucleosome cores that are in association with histone 1. It holds the two cores together. So you require the linker DNA, you require H1, you require H2, A, H2B, H3, and H4. Fine? And the linker DNA. Now, another interesting thing is here, the tails. Tail of H2A, H3, and H4 interact with the adjacent nucleosome. They get attached to the adjacent nucleosome. High compactation of DNA involves large loops of nucleosomal DNA. Now, histone variants alter nucleosome function. Now, what happens is if they become very tightly condensed together, they become very tightly condensed, they will become, the genes become silent and they become repressed and they become heterochromate. You remember? But you don't want that to happen all the time because many genes have to be translated. So it has to be loosened up. If an H2AZ histone inhibits nucleosomes from forming repressive chromatin structures, creating regions of easily accessible chromatin that are more compatible with transcription. had muted it. Sorry, I answered my phone. That's why there was no sound. So H2AZ histone inhibits the nucleosome from forming repressive chromatin structures, creating region of easily accessible chromatin that are more compatible with transcription. So these H2AZ break it up a little bit so that the DNA, the transcription factors come there and RNA can come and DNA replications can take place. Now, nucleosomes that include some nucleosomes have to be included into centromere. For that, you have the centromere protein A attached to the nucleosome. One end will join with the kinetochore protein. Okay? So what happened so far? We talked about H2A, H2B, H3 and H4, how they will form the nucleosome. Then once they form the nucleosome, the H1 will come as a clip and sit on it and make it tight. Now, and also a linker DNA is also needed to link the things together like a string. Now, then it cannot be too tight. If it becomes too tight, it gets repressed. So to prevent that, you have the H2AZ histone. When that comes in, it inhibits nucleosome from forming very repressive chromatin structures. Then you also have a region which has to get associated with the centromere, so you have CENPA. Okay? Clear? Now, the interaction of DNA with histone optimer is dynamic. Why is it dynamic? Why is it dynamic? Why is interaction of DNA with histone optimer dynamic? Oh, no, dynamic means what? What does dynamic mean? What does dynamic mean? No. The word dynamic means what? What is the meaning of the word dynamic? Look it up and tell me. Okay, Baba. Dynamic means it is changeable. It has to be able to change. It cannot stay stable. If it stays stable, it cannot change. Means if it sticks together and the association is very tight, there is no translation. So dynamic means something which can change. Okay, it doesn't have to be in one spot. Like we always have to be dynamic. Allah wants us to evolve. Allah wants us to be better than yesterday. So we have to be dynamic personalities. We should not be placid. We should not be static. Okay? So, fine. Now stop writing. It's okay. So it has to be dynamic. The interaction has to be dynamic because it has to unwrap, it has to wrap, a translation has to take place, mitosis takes place, it has to condense, it has to open up. So it's constantly changing. Means every cell in our body is like a universe. It's constantly changing. Unwrapping of the DNA from nucleosome is responsible for accessibility of the DNA. you have a protein binding site 1 and a protein binding site 2. Frequent event, protein 1. Protein 1 will bind and then it will unwrap, or sometimes protein 2 will bind and it will unwrap. So it is a dynamic structure. Nucleosome movement by nucleosome remodeling complexes. Now, how does it remodel? We talked about transposomes, genes will jump, but here it doesn't jump because it's like a bead on a string. So the movement is limited. It can either slide, it's like a sliding, see? It can slide and the proteins can come up. Sliding remodeling, see if I tell you the kinds of nucleosome movement, nucleosome remodeling, name them. One is by sliding, one is transfer, just transfers it out, or restructuring. The inaccessible one can become accessible. Okay, so the remodeling, the movement takes place by either sliding, the histone will slide, or it will get transferred, or it will get restructured. All these require energy, okay? Energy from ATP, energy from all of them are dependent on energy. So some source of energy has to be there for the hydrogen bonds to break. Now, ATP-dependent chromatin remodeling complexes. What is a bromodomain? Please check out and tell me bromodomain. What's the chromodomain? You can Google search and tell me then what they are. What is a bromodomain first? Amino acid, what kind of amino acid is it? Amino acid pairs, not an amino acid. Amino acid base pairs. Okay, approximately 110 amino acid proteins that recognizes acetylated lysine residues. Very good. Where are the lysine residues? Where are the lysine residues? Can you tell me? Where are the lysine residues? Terminals of N-terminal of the histones, right? Very good. So the bromodomain will recognize that. SW1 and SNF. What does it stand for? Take it, get it for me. There are 8 to 11 subunits, so it's quite a big thing, remodeling complex. ISW1, 2 to 4 subunits. No, it's not a bromodomain. What is a chromodomain? 8 to 10 subunits. Chromodomain 42 commonly found in proteins which associate with. Okay, of amino acids. Very good. Of chromatin protein structures. Okay, so we got this. Now, what is SW1 and SNF? Can anyone tell me? SW1, SNF. Okay, good, good, very good. All these will become in your quiz also, huh? What is your SW1 SNF? I know, but what does the name stand for? SW1 SNF. The name. You should try to remember what the name stands for. Otherwise, you forget it, you know. But if you know what the name stands for, you remember. What does the name stand for? Good, I'm glad everybody's participating today. Oh, very good. Switch sucrose non-fermentable. Very good. That is what it is. What about ISW1? Which means switch sucrose non-fermentable. This is the name of the gene. ISW1. Anyone can tell me? I can't find it. Anyone found it? I can't find it. I just guess. Oh, yes, I found it. It's a dream, okay. But the name ISW1 stands for? Can anyone find it or should I write it? I just found it myself actually, but I'm Googling it just like you. So this was SW1 SNF was switch 1, non-fermentable, it's a bromodomain. IS1 is, ISW1 is, they're all chromatin remodeling complexes, okay? No, no, no. Imitation plus chromatin remodelers. So IS1 means imitation switch 1. Okay? What about MI2? MI2 NURD? Come on. NURD is obvious. Nucleosome remodeling. Nucleosome remodeling deacetylase and deacetylase complex. But MI2, why they have named it MI2? I know. Okay, Mi2 nuclear remodels, I know. No, I just wanted to know what the name stood for actually. But I can't find Mi2. NURD is nucleosome remodeling deacetylase. Deacetylase, yeah. Deacetylase complex. Nucleosome remodeling deacetylase complex. Yeah, I know. No, I just wanted to know what the words, the names came from. Like SW1, SNF means switch 1 non-fermentable. So we remember that as a bromodomain, switch 1 non-fermentable, SW1, SNF. ISW1 we said was imitation switch 1. It's not a bromodomain. This is nucleosome remodeling deacetylase complex. Mi2 I did not find, which is a chromodomain. Okay, that's good. Very good. So these are all ATP dependent. ATP to give them energy. Not coming? No. Nucleosome positioned by DNA binding proteins. What were the DNA binding proteins? SW1, SNF, then NURD, MI6, and the other one was what? The third one? Imitation IMI, ISW1, imitation switch 1. These are all the remodeling complexes. Now, positioning is this. First, an exclusion can take place. This protein, this DNA can go out. This is your protein which gets bound, protein B. This place becomes nucleosome free. So this becomes nucleosome free, your RNA will come and bind, your transcription factors will come here and they will bind. So first, a factor will have to come to remodel your nucleosome. So the nucleosome opens up and part of the DNA gets exposed. It becomes nucleosome free and then your transcription can start. It's very beautiful, isn't it? Really beautiful, really beautiful. The first one was exclusion. It excludes. Another one is inducing assembly. Now you've got it, now you have to get positioned by DNA binding proteins. This is your position, nucleosome will come. This will come and bind it. Okay, so you have exclusive, then you have inducive. Now, modification of the N-terminal histone tail alters the function of chromatin. You have a histone fold. K is your lysine. What does S stand for? Can anyone tell me? What is S? Sustain. Yeah, yeah. Oh. Serine. Serine and lysine. You remember we said serine, S is serine and K is lysine. So your serine and lysine are very important. For yellow is phosphorylation, red is methylation, blue is acetylation. This is your histone fold. Acetylation, your transcription is activated. So you have acetylated lysine. Here you have methylation, then you have phosphorylation on the, what was this we said? Serine. Now, gene silencing, gene expression if acetylation takes place. If acetylation is on lysine number 9, histone deposition. If phosphorylation takes place on number 9, number 4, lysine, gene silencing, heterochromatin gets formed. Here, condensation. Gene expression. Transcription elongation. So, effect of histone tail modifications. You remember we said when they get acetylated, H2BH3, the transcription starts, the bromodomain proteins will come. Once the bromodomain proteins will come, remodeling takes place. It will open up and remodeling takes place. If they're methylated, gene side, the chromodomain proteins will come and bind. What was the chromodomain protein, if you remember? Chromodomain was the ISI, right? Sorry. ISW, no, sorry, Mi2NURD is the chromodomain. The chromodomain, the bromodomains will give activation, chromodomain will give repression. Now you have your enzymes. A lot of enzymes are there. Histone acetylase transferase complexes. They're also bromodomain or chromodomain. Saga, the type is Saga, PCAF, and you should remember at least one or two of the names, okay? The catalytic subunits, bromodomain, you should remember that one or two of them, one chromodomain and one bromodomain. We'll ask you to name it, NUA4. Histone deacetylase complex, SYN3 complex, NURD, SIR2. They are all the enzymes for deacetylase. Histone methylases, chromodomain is a neither. The target is lysine 9, lysine 4, arginine. They become methylate. Once they methylate, what happens here? Methylation results in what? Does it result in transcription or suppression? What does it result in? Suppression, good. The spelling is not like that, huh? Okay. Acetylation activation. Deacetylase also suppression. So chromatin remodeling complex and histone modifying enzymes work together to alter the chromatin structure. DNA binding protein, chromatin remodeling complex, the fibers. the histone acetyl transferase will come. It will open up. Here, DNA binding protein 2. Okay, I'll stop here. I'll give you a 10-minute break. Wash your faces, walk a bit. Okay, and we'll come back at exactly 12:30, inshallah, where I'll give you your quiz.